Good afternoon. Thank you, everyone, for joining us for today's Research and Innovation Training Institute. Today, our guest is the Director of Research Compliance, Donna Pulkrabek. Donna has been with Prairie View A&M University since 2015, and we are excited about learning more about research compliance and biohazard safety. Donna, thank you for joining us.
Thank you. And I think I'll just get started. So, hello, everyone. Thank you for coming. As Ms. Cotton said, my name is Donna Pulkrabek. I'm the Director of Research Compliance, and I'm going to talk to y'all today a little about biohazard research ethics. So, without further ado, how do we define research ethics? I think everyone has their opinion about what ethical behavior is, but when it comes to research, we have certain ways of thinking about it. So first, I'll kind of describe to you the human world, then the animal world, and then we can dive into biohazards. So in the human world, when we do human research, we generally have three concepts we're looking for ethical behavior. Do you respect the person, meaning their right to choose whether to participate in the research? If they have diminished autonomy, are you protecting them? You'll think of beneficence as well, meaning don't do any harm, and maximize the benefits and minimize the harms if there are any. Justice, if you receive the benefit, you should bear the burden as well. In other words, don't do human research on one group because they're convenient and then let another group benefit from it. So the solutions for humans are things such as informed consent, voluntary participation, assessing risks and benefits of the research, and equitably selecting the subjects. Now in the animal world, it's a little different. An animal cannot sign a consent form, obviously. So we think of the three R's. You can refine experiments by minimizing pain and distress. You can reduce the number of animals you need, and you can replace animals whenever possible with the alternatives. So solutions would be doing fewer procedures, doing established experimental endpoints, such as when the tumor reaches a certain size, we shut down the experiment, we don't let them suffer beyond a certain point, using statistics to calculate the minimum number needed of animals, and you could use computer models or tissue culture instead of animals. So that being said, how do we define biohazard research ethics? So it's not a human and it's not an animal. So what do? Well, we can start by asking a few basic questions. So you don't have to type anything in the chat, but just think about these and I'll just kind of expound on them. Is it ethical to expose your coworkers to a laboratory-acquired infection? Now, I think most of us would say, no, that's not very ethical, Donna. And I would agree. Now, there is a certain amount of risk with everything we do, but for you to carelessly expose your coworkers, it's not good. Number two, should personnel know the health risks of working with a particular agent before they start work? Well, going back to the human research, yes, they need to know what they're getting into. So I think most people would say yes. Number three, should those who enter your lab be informed of the risks/agents present before they enter? Yes, I think most people would say yes. I'd like to know, is there a sign on the door? Most biohazard researchers have a big biohazard orange sign that has the names of the agents. So you know what you're potentially exposed to if you go in that room. Can some infectious agents be used for military applications? Well, definitely. But yeah, we've seen that history. I'll talk about that in a minute. So is it ethical to make a pathogen more infectious? And I think gain of function is the term that's used. Yes and no. But if you think about it, are you using it? It's kind of like a knife or a gun. A knife or a gun inherently aren't bad. It all depends on how you use them. You can use a knife to cut up your vegetables or your dinner. You can use a gun to protect your home, but you can also use a knife or a gun to hurt somebody. So, you know, it's kind of a, you know, what are you going to do with it? Same thing with the pathogens. So have there been attempted murders in research labs using biotoxins? And yes, there have been. I have actually heard from a PI that's had 2 in her lab. So yes, it does happen. So people are people, tools are tools, and biohazards can be used in a bad way for bad things. Last question, can you destroy an ecosystem by the release of a recombinant agent or a biohazard? And that's an absolute yes. So let me take a theoretical example. Let's say you were in your lab and you were researching black spot that, you know, on citrus, when you buy your citrus and you're really upset because you get them home and they have this big black spot on them and it's nasty with fungus and you have to throw it away. Well, what if you took that fungus and you did some gain of function research on it, made it more pathogenic, and then you accidentally released it in South Texas? You could destroy the entire citrus production in South Texas permanently, or for decades or however long until it could be eradicated. Take it to California, take it to Florida, you've destroyed the citrus industries there. So yes, definitely you can. So bioethics is thinking, I would say it's like thinking about what you're doing and the possible consequences of what you're doing. Okay, so let's talk about laboratory-acquired infections. This is a true story. In 1972, a researcher was working with a rabies virus. This researcher was immunized against rabies and was processing a 5-year-old fixed goat brain tissue. Now, this goat brain tissue was infected with rabies. And what this researcher did is he put the brain tissue in a kitchen blender and turned it on a bench top, not in a biosafety cabinet where the air protects you. Did it in a bench top, takes the lid off, sniffs it. This is the researcher's regular practice, sniffs it, and uses a mouth pipette, like a straw, for those of you who don't know, to transfer this. There was no skin puncture, no splash. He died. So what happened is he aerosolized rabies, breathed it in. Now, most likely is what happened. So how could this have been prevented? Y'all think to yourself, how could this have been prevented? Well, one, since he was immunized, he could have gone and made sure he had a titer against rabies at regular intervals. That wasn't being done here. So yes, you may have had the vaccination, but that lasts for a predetermined, usually number of years, and he didn't have protection anymore. He could have been doing this blending inside the biosafety cabinet to minimize aerosols. Anytime you're blending or pipetting, you should do it in a biosafety cabinet just in case something gets aerosolized, that air barrier protects you. I hope I don't have to tell anyone that you shouldn't be sniffing anything in the lab or mouth pipetting. These are very dangerous, but this researcher was doing that. And he was using appropriate, apparently when this sample of these goat brains was fixed, the chemical was not appropriate, I guess, to deactivate the rabies virus. So clearly it wasn't, because it's 5 years old, it's blended on a bench top, and he gets rabies from it. So that's one of the things the IBC looks at, is whether the chemical you're planning to use is appropriate to fix or disable, decontaminate the pathogen, the agent we're working with. So congratulations. If you were thinking about any of these ways this could have been prevented, you are now using biohazard research ethics skills. because that's really what it is. It's common sense. And a lot of it is just flat common sense, but with a little bit of scientific knowledge, of course, but that's a lot of what we do. Let's talk about another one. 1997, Shigella, a student working in a clinical lab, used the hand washing sink to clean and decontaminate labware, like flasks, tubes, things like that, because the dedicated sink was full. So this student likely contaminated the sink faucet handles. Everyone who worked in the lab, there were 7 other people in the lab. The student did not get sick because they were wearing gloves. 6 of the 7 people in the lab got sick. The 7th person did not. The 6 people who didn't get sick were using their bare hands on the sink handles that were contaminated by the student. The one researcher who didn't get sick used paper towels to shut off the handles. And Dr. Whitney tells us many times that's exactly what he does. He's the chief research compliance officer for the system. So something to think about. So how could this have been prevented? Okay. Training. Could have trained the student a little better. Don't wash dishes in the hand washing sink. It's dedicated. You could have used disposable labware so you didn't have to wash it. You could confine the work to the biosafety cabinet, including the waste collection. And they could have hands-free sinks, you know, the ones that use the foot pedals. And that could have done everything. That could have not done everything, but it would have drastically removed the risk. By the way, can anyone tell me what's wrong with the picture? Anybody? She's not wearing gloves.
No gloves?
Yeah, bingo. What I like is whenever I bring that up in a training class, it's never the scientists who say they're not wearing gloves. It's always a non-scientist that tells me that. So I'm like, yes, employ common sense. This is what research ethics is. You think before you do. So let's talk a little bit about military applications or terrorist applications. So here's a few historical examples that I found online. 1500 to 1200 BC, the Hittites drove victims of tularemia into the enemy lands to cause an epidemic. Tularemia is rabbit fever, highly infectious. In 300 BC, Scythian archers, they dipped their arrows in decomposing human cadavers and snakes or blood mixed with manure to cause tetanus. So it's bad enough you get shot by their arrow, but then you're going to probably die of the infection you get because they took their arrows in this. 1346 AD, the Tartars hurled plague-infested bodies over the walls of the city of Kaffa. I believe that's in the Black Sea, to hasten the surrender of the city. So those are some historical examples. Do y'all know that there has been a bioweapons attack on US soil? I didn't know if y'all knew that. September of 1984, there's a local religious cult in Vales, Oregon, who wanted to sway the results of an election in favor of their candidates. So they were doing a trial run. They were going to do this right the day before the election. They sent their cult members to several local salad bars and sprayed the food with salmonella. They got 751 people sick. Fortunately, it wasn't a highly dangerous strain of salmonella, but yeah, I got a lot of people sick, and I believe the FBI kept this quiet for 20 years because they feared a nationwide panic if it was widely known. But I think they finally released it in the 90s. All right, so that's an example. So why am I bringing all this up? Do you keep your lab locked? Are your freezers locked all the time? I mean, think about that. There aren't nice people in the world. If they got a hold of your samples, even if it's maybe not a highly infectious sample, they could harm or kill people. The restaurant owners, many of them went out of business here. I mean, they lost their livelihoods because of this. So don't think it can't happen again. All right. Okay, so let's talk about ethical principles for biohazard research. And here's just a few. This isn't a comprehensive list. It's just some for y'all to think about. Don't expose people to a laboratory-acquired infection. So do a risk assessment and mitigate the risks. Inform personnel and visitors to the lab of the risks or agents present. So you want to train your personnel on what to do and not to do, and inform visitors. The signage is part of that. Sometimes you need more. Number 3, can the infectious agent you're planning to work with be used as a weapon to living organisms or ecosystems? I don't believe anyone here works with DIRC agents. That's dual use research of concern. So that would definitely include all Rg4 level agents. Those are your Ebolas and, Black Death, Yersinia pestis, those kind of agents, Marburg virus. But you can always contact us if you're not sure, the IBC, Institutional Biosafety Committee. You can do a risk assessment and mitigate the risks, and of course, always review your lab security. By the way, in the picture, did anyone catch that the Mona Lisa is wearing a mask? I thought that was really great. Okay, so let's talk about the risk groups and biosafety levels. Risk group and biosafety level are two different things. So a risk group is, risk groups are found in the NIH guidelines. I'll cover that in a minute. That kind of tells you how risky your agent is, going from a 1 to a 4, and I'll talk about that in a minute. The biosafety levels also go from a 1 to a 4, but they are basically describing your containment levels, like what practices and what equipment you're using, what PPE is required. So generally speaking, the risk group and the biosafety are usually the same, but that's not always the case, because you could have something that is considered a risk group 1, but you've made it a little more dangerous, so you need to use it at a biosafety level 2. And that does happen. So going to this. Risk group 1, they're often used in BO1 labs, but they don't cause disease in healthy adults. It's, for example, maybe most animal cell lines. And the general statement thing to think of is don't drink it. So I think of Rg1. When I think of Rg1, I think of this office I'm sitting in, or just maybe a teaching lab for undergrads. Okay, Rg2 agents are often used in BL2 labs. They can cause infection of varying severity, rarely lethal. Treatment's usually available. Example is Salmonella, Listeria, agents like that. And the general rule of thumb is don't touch it. We have some BL2 labs here at Prairie View. Rg3, risk group 3, is often used in BL3 labs. These are agents associated with moderate to severe disease, can be lethal, treatment may not be available. So, such as tuberculosis. For those of you who know your history, there were many sanitariums in this country. In the early part of the 20th century, many people died until the advent of antibiotics. There weren't many ways to treat tuberculosis. So the general rule is don't breathe it. And I have inspected these kind of labs before. They typically have you wear a full PAPR, which is the airflow, brings in air, blows it out, so that air doesn't get up inside your face that has the agent, the tuberculosis in it. So you're basically, as you're moving up in numbers, you're moving up in danger. And then Rg4 is used in BL4 labs. I can't think of an instance where it wouldn't, but that is capable of causing severe disease with lethal outcomes. Treatment's not generally available. So now you're thinking of Ebola, Yersinia pestis, which is the Black Death, those kind of agents. And the statement is don't do it. So 1, don't drink it. 2, don't touch it. 3, don't breathe it. 4, don't do it. There's only a few BL4 labs in the world that we know of. I hope there's not more. But yes, this is for the very dangerous agents. Okay, so how do we prevent people from getting sick? The two most important concepts are risk assessment and containment. So we need to think, what are the agents we're using and how are they being used? Because that's going to tell us what to do with risk assessment and containment. Okay, for risk assessment, let's think of the chain of infection. So let's say you have a BL2, let's say you have salmonella in your -80 freezer. That might be the reservoir of the pathogen, right? That's where it's stored. So what is its portal of escape? You take it into your lab and you open the tube, right? It can now escape. So how would I mitigate that? Well, you'd mitigate the risk with your practices and your equipment. Don't open the tube on the benchtop. Open it in the biosafety cabinet only. Use a biosafety cabinet, right? That's equipment. Make sure it's running. Your biosafety cabinet is operating. Your SOPs, you're using gloves, right? Well, that's actually the next one. But your practices and your equipment help you in your first line of defense. So the next thing that would happen is it would be transmitted to a human. So how do we mitigate that? Well, you have your gloves, you have your mask, you have your smock. These things you wear, maybe eyeglasses, that's going to drop your risk. If you splash it, you drop it, splashes on your eyeglasses, at least it's not splashing in your face or in your eyes. So that's another level of protection. Well, let's say it passes that and you get a route of entry, an infectious dose. Okay. Let's say you cut yourself with a sharp or, it splashed in your mucous membranes because you forgot to put your glasses on. Next, you need a susceptible host. So how do we not be susceptible? Well, immunization, if it exists for whatever you're working with, that's helpful. And make sure your titers are up to date if necessary. So that will hopefully stop it there, right? Because you're immunized. But let's say you're not, and you hit the incubation period. Okay, what's next? Well, surveillance. So you might be thinking, well, okay, why is this relevant? Well, this is why we ask you to call OC Health immediately as soon as you get medical attention, because they're going to tell you what to watch for. Like if whatever you accidentally might have infected yourself with is going to give flu-like symptoms, they'll probably tell you to stay home and for a few days and call us if you have flu-like symptoms. Don't go anywhere. Have your food delivered or something. So that's basically the chain of infection. You have to cross all those barriers to get sick. And the IBC, the Biosafety Committee, is looking at all these and the OC Health program to prevent that, hopefully. So what's the next level is biocontainment. So how do we contain your agents? We use practices, PPE, which is personal protective equipment, your lab equipment, and your lab design. So let's talk about the 4 different types of labs so you get an idea of the kind of equipment. BL1, that could be just gloves and lab coat. You've got a sink and a door. Just your generic teaching lab. A 2, you might have safe sharps practices, base protection, a biosafety cabinet, the removable rotors from your centrifuge. You take it and unload it in the biosafety cabinet. Controlled airflow into the lab. So if you ever see me with a little piece of paper at your door, I'm seeing if the air is coming in, not going out. So if it's going out, it could be blowing your agent out in the hallway, and we don't want that. Number 3, okay, all work done inside a primary containment device, so no benchtop work with an agent here. Add respiratory protection if needed. I've always seen it used in a true 3. Add a dedicated HVAC system with no recirculation of exhaust, so you can't have a biosafety cabinet that recirculates air in the room and has to go out the building. 2-door separation from the hallway. No one can stumble in. You need to have lots of security. Fan failure alarms and HEPA filtered exhaust, in case something gets out in the exhaust, if that's needed. And a 4 is adding a supply airline respirator. You've seen the movies where they have the big white suits and the headgear, and that's what we're talking about here. So you want a supply airline respirator, fully encapsulating protective suit, all work in sealed boxes. It's like a building within a building. And all systems for lab are separated from the non-BL4 areas. Wear a DUBA HEPA filtered exhaust air, HEPA filtered supply air, and an effluent decon system. So that's not for the faint at heart to work in these places. I know that Texas A&M has some 3s. I don't think anyone at Texas A&M has a 4. Correct me if I'm wrong, anyone. But. Okay, so where can I go for more information on the biosafety level for my agent and the recommended precautions, right? I'm going to give you 5 resources, and you go there in this order, because this is how the IBC is going to review your documents for your ethics approvals. Number 1, go to the USDA APHIS Agricultural Select Agent Services, AgSAS, and the Health and Human Services Select Agent Federal Regulations. And there's the link. Odds are you're not on this list unless you're using a select agent, but it's always good to check, because that will be the number 1 thing precedent-wise. Number 2, go to the NIH guidelines. And this is on NIH's website. You'll need this definitely if you're doing anything with DNA, you're manipulating DNA. You definitely want to go to the NIH guidelines. This is also, there's an appendix to these that have all of the risk groups that I just talked about. So if you want to know what your risk group is, try the appendix here. It's probably there, even if whether you're doing DNA or not, whether you're manipulating the DNA or not, it should have your risk group. Next level, the BIMBL, Biosafety and Microbiological and Biomedical Labs, 6th edition. It's most recent. This is where you're going to find your biosafety level. They have like specific agents. So if I went and looked for brucellosis, I'd go in there and I'd look up brucellosis, and it might tell me you have to use it at a BL2. You know, BL2 is what you should use. So that's where you'll get some information on biosafety. It also lays out in detail what your biosafety level lab should have in it, like what I just described. Number 4, the American Biological Safety Association Risk Group Database. This is free. You don't have to be a member. You can go there and look up your agent and find out what they say, what they recommend for containment levels. Could be a 1, 2, 3, 4, depending on whatever it is. And the last stop, if you haven't been able to find what you want, go to Public Health Service Canada, the Pathogen Safety Data Sheets. It's really nice. Canada has some lovely data sheets on many different pathogens, so you can maybe find it there. All right. And this is, like I said, what the IBC is going to use to judge what you're doing. So when do I need this IBC protocol approval? Okay. And a protocol is, by the way, a document describing what you're going to do with the agent. So that's what the IBC committee reviews and approves. So you'll need it if you're going to do work with anything infectious. Infectious to humans, animals, insects, plants. If it's infectious to anything, the IBC needs to see it. If you're using human tissue or cell lines, definitely, because that's infectious to us. If you're using recombinant DNA, manipulating DNA or RNA, definitely. Now, that doesn't just simply amplifying it as PCR, no, but if you're splicing, cutting, changing, yeah, you need it. Biotoxins, if you're using toxins originating from a biological origin, like from an animal, such as snake venom, or a plant, such as ricin that comes from castor beans, you do need an IBC approval. If you're doing plant research that requires a USDA permit, definitely, because we go and just check and make sure the permit, you're doing everything you need, got everything you need for the permit. Situations in which you might need an IBC protocol, if you're teaching like a undergraduate biology or food science courses, yes, you'll need some sort of approval. Right now, that's with the IBC, so call me. We'll get you taken care of. It's a different form than the usual protocol forms. It's teaching. If you're doing animal tissue or cell line work, it depends, and I'll talk about that in a minute. So here's the it depends. Do I need an IBC or an IA code? Good question. If you're using animal tissue or animals, which approval do I need? all activities involving live animals in their naturally occurring state. They might be sick, they might have a zoonosis, like the goat might have brucellosis. That's IACUC only. Okay. If you're in a naturally occurring state, it's okay. Just IACUC. Don't need the IBC. All activities involving the deliberate introduction of a biohazardous material, such as an infectious agent or recombinant or synthetic DNA, RNA into animals, you need both. Go to the IBC and the IACUC. If you are extracting a select agent from a naturally occurring infection of animals, so for example, you went to that sick goat and you were trying to get brucellosis, a sample of brucellosis out of him, or rabies from that previous example, you need both. You need IBC and IACUC. And for most people, all activities, research, teaching, demonstration involving the handling and manipulation of animal tissue in vitro and other biological material from animals, we need to do a risk assessment. The IBC needs to do a risk assessment to figure out if an approval is needed. Sometimes it is, sometimes it isn't. Once again, it's agent specific or risk specific. For example, with goats we use, their reproductive tissue is much riskier than, say, muscle tissue. It's more likely to contain brucellosis, which can cause abortions in women. That's why the tell us, there's a short form I'll send you. Send it to the IBC. The IBC will do an assessment and let you know. All right. So what if I need both, IBC and IACUC? Get the IBC first. That's the thing. Get the IBC first. Start the paperwork with the IBC first. You can go ahead and prep your IACUC, but the IBC is going to take longer, and that's the first one you should do. It just makes more sense to do it that way. Okay. So I want to start work, Donna. How do I start work with my IBC protocol? Well, there's three things you'll need to do in the IBC world. You need to get your protocol approved, you need to get your lab certified, and you need to go through OCHealth. So, protocol approval, you need to send us the document. I can send you the documents that you need to fill out. It's a 4-part form. You may not need all of them. And then your lab SOPs. And you'll need to have everyone finish the trainings. I'll talk about those in a minute. Lab certifications, it could be a 1 or a 2 here. We don't have any 3s. In-person, we'll be doing an in-person inspection before you're certified, and then there's an annual re-inspection. And OCHealth, it's initiated only after the protocol is approved. There is a training component to it, which my office sends out, and then there's an enrollment component which risk management safety handles. That's the medical questions, should you choose to answer them. And then we'll annually ask you to re-enroll. Now, as far as training goes, there's 3 types of IBC training you may or may not need. There's your basic biosafety training. That's everybody listed on any protocol, 1 or 2. It's online, and you retake it every 3 years. Then there's the BL2 lab training. That's for all personnel listed on 2 protocols, and all personnel working in a 2 lab, regardless of their job duties. So if you have a student that happens to be working in your BL2 lab, but they're using BL1 agents, they still have to go through this because they're potentially exposed, right? Because we're going to need to tell everyone what they're exposed to before they walk in, right? Because we're ethical, right? We don't want anyone being sick. This one's available on TrainTrack. Normally, it's given in person, but because of the pandemic, we've been accepting TrainTrack. And you retake it once a year, as long as you're working in there. NIH guidelines is just for the principal investigators. It's in TrainTrack, and you retake it once a year. All right. Questions? That was everything. Anyone? Did I put y'all to sleep? I'm sorry.
We're still here.
Okay, good. All right, well, thank you for your attention. My name's Donna, and this is my contact information. I see something in the chat. All right, thank y'all. Thanks Thank you, Dean Luter.
We do have a couple of questions, Donna, before
Okay.
we close. So one of the questions is, if you could go into detail on this, I know you kind of touched on it, but what is an IBC protocol?
Okay, a protocol is a document that you send to the IBC, which is the Biohazard Ethics Committee. And this document tells us what agents you're working with, what you're doing with them. Like, you know, if you're passing cell lines, if you're, you know, doing translation or transfection, you know, you're putting DNA in something, taking DNA, whatever you're doing. It also tells us about your decontamination procedures, you know, where your autoclaves are, who's working with you. And we'll ask for your SOPs, which are just generic. Here's how I clean up the spill. Here's what we do when things happen. Here's security, you know, just your general practices.
Okay, and if there's something that needs to go through the IBC process, how often does the IBC meet?
Well, we're scheduled to meet the second Friday of every month.
Okay.
So, yeah, all new protocols go to the committee, all 30-year renewals. Every 3 years, we ask for your protocol again, and we renew every 3 years, or review for sure every 3 years. There's an annual form, but if you're not making any changes, we don't generally take it to a meeting.
Okay, and you mentioned several training classes. How can a person get information on the training classes? Who do they contact?
Okay, you can email me. My email is on the screen. It should be on our website as well. I believe it is last time I checked. So all you have to do is go to the main webpage, go to research bar, and then you'll see research compliance, and then, or you just type IBC, you should be able to get to it. IBC forms.
And the same is true for lab inspections and protocol. Any of the
protocol? Call me or email me here. Our research compliance email does still work. I'm answering all those right now. So.
And I want to remind everyone that today's session will be available on our YouTube channel, which is PVAMU Research and Innovation, our YouTube channel. And please subscribe so that you can get any of our trainings or any of our videos whenever they are uploaded onto our YouTube channel. Donna, thank you so much for the information that you shared with us today.